Thank you, and thank you for inviting us and giving us a platform, and being called one of the flagships of IMI is really an honor. We were the first IMI 2 project to go through, and so we went through the birth canal, if I may call it that way, and so we had quite a few hiccups on our way, but we plowed the way for all the projects coming after us. So just to situate, what is INNODIA? INNODIA is really a project on innovative ways of trying to understand what is type 1 diabetes, and also finding biomarkers, getting us to a prevention and a cure eventually, hopefully, for this disease. Most of you will know diabetes, but very few will realize that type 1 diabetes is actually a very small part of the big epidemic that is type 2 diabetes. That is diabetes. The big part is type 2 diabetes. So when we talk to pharma companies, when we talk to policymakers and we talk about diabetes, and as a clinician I see a lot of people with type 2 diabetes, everybody gets very excited because they know these figures, and this is in the meantime a very old slide. But when you say, yeah, but it's type 1 diabetes, they go, oh, you know, much smaller market. But still, there are companies out there who really want to make a difference. Now, what is type 1 diabetes? It's very different from type 2 diabetes. Type 2, mainly a disease that has to do with overweight, with not moving enough, with also becoming older. Type 1 diabetes is an autoimmune disease where one cell in the body, just one cell, the beta cell, the insulin-producing cell in the pancreas, is being destroyed by the immune system. And so therapy really consists of replacing all the functions of this beta cell. And this beta cell is one of the most differentiated cells in our body. It's one of the most specialized cells in our body that measures continuously our blood glucose, then has an algorithm and secretes insulin directly into our blood, and is able to really maintain our blood levels at a very stable level, whatever we do, whether we eat a lot of cakes or whether we fast for 3 days. And so this is the challenge for our patients with type 1 diabetes, namely they need to replace all these functions of the beta cell externally. So they need to measure their glucose, they need to reflect, and then they need to administer insulin. And so this is really what we're doing. We're trying to replace, all these functions. Now, this is reality, namely, type 1 diabetes is a disease that will typically also affect children. And so if a child is diagnosed when she's 5 years of age, it means that she will have to dramatically change her lifestyle and start to prick herself, start to inject herself insulin, and that for the rest of her life, in order to maintain good blood glucose levels and in order to avoid complications. So people who work with the disease that is type 1 diabetes really are very close to patients. And so it shouldn't be a surprise to you that the very first project to go through and the very first call to be launched within the IMI 2 was on a disease that is type 1 diabetes. And it is a call where FPA partners came together. It was Sanofi, Novo, GSK, and Eli Lilly. But to be very honest, this call was realized through the patients. Namely, JDRF, the Juvenile Diabetes Research Foundation, helped also by another foundation, Helmsley, really were the ones who drove this call. So this call originated from the patients because they said, we need to do things differently, we need to be disruptive and really get to know this disease that is type 1 diabetes and get to a solution. Because this is what is the challenge. Namely, our patients need to maintain their blood glucose levels, and everything has an influence. Stress has an influence. What they eat has an influence. And so this is reality. This is a continuous blood glucose monitor showing day-to-day variations in blood glucose. And so finding ways to really prevent this disease and cure this disease would really be disruptive. And at the basis of being able to prevent this disease is still better understanding of the disease and also getting novel biomarkers. And so this is really what INNODIA is about, to come to translational approaches to disease-modifying therapy of type 1 diabetes. We called it an innovative approach towards understanding and arresting type 1 diabetes. So this is the group. It is a big consortium where, as I said, we have 4 FPA members, but also we have these 2 foundations. JDRF, actually, it originated from parents of children with type 1 diabetes, and the Helmsley Trust, 2 foundations really driving this initiative. And then we have 27 academic partners and also an SME. Actually, by the time we started INNODIA, the SME was already taken over by another SME. So we had our first amendment to be done anyway. So, yeah. So if you look at our abstract, this is it. Don't read it. I put it for you into a word cloud. And so if you put this abstract into a word cloud, this is what you get. Namely, what is INNODIA about? It's really about a combination of basic translational researchers and clinical researchers. It's a combination driven towards interventions, clinical interventions, doing trials, and having patients in the middle of all of that. So, thinking again about the topic of today, open innovation, how is INNODIA novel? How do we do things differently? Where does this innovation come in? So this is our organization. You could say, well, this is standard work packages, advisory boards, and what have you. But let me show you some of the things where we think we are different than what other people have done. A first work package is really clinical, and it is the establishment of a network throughout Europe of sample collection in a standardized way. Not very scientifically exciting, but crucial. If we want to understand type 1 diabetes, this is what we need. Careful clinical phenotyping of our patients, or also people at risk of type 1 diabetes, and then a standardized way in collecting samples. We collect urine samples, blood samples, stool samples. We foresaw 3 years ago that microbiome would become big, and hey, it has become big. So we have stool samples being collected. We hope for a restricted call to be able to analyze them, but we still need to get there. But so a standardized way of collecting samples. So this is the way it is. It is a huge network. It is 17 clinical partners where we aim to collect 1,500 newly diagnosed type 1 people and also 3,000 of their unaffected family members. Work package 2, what is new there? It's the technology hub. You can say, well, that's not very novel. I can tell you, this is disruptive. Don't read it again. It's just to show you that for the first time in history in Europe, and I can tell you in the world, we do a standardized suite of analyses. All the autoantibodies are being determined in one place. All the T-cell assays, the assays of the immune cells, are being done in specialized centers, all using the same standardized protocol. Again, very boring, but so crucial to really be able to come to a very solid conclusion. Work package 3, basic research. Not very novel. Everybody does basic research in type 1 diabetes, yes, but this is now basic research and translational research really focused on the discovery of novel biomarkers of the disease and also novel biomarkers of therapeutic success of some interventions. With, and you see the arrows, with the immediate possibility of transferring interesting novel biomarkers to work package 2, so immediately also testing them in the samples that we have gathered from the patients. And I can tell you, we've started in January, it will be 2 years now that INNODIA is really operational. I can tell you that we have the first biomarkers being transferred from work package 3 to work package 2, namely a set of microRNAs. Fortunately, we built in standardized ways of collecting blood that is able to allow us to analyze microRNAs in the blood of our patients and first-degree relatives. Not a trivial feat, because literature is really littered with reports on microRNAs in people, but if you use plasma or serum, it's very different. So here, everything is collected in the same way. And then perhaps the most crucial part, that is the fact that all the data, not only the data from the patients, from the first-degree relatives, but also from our in vitro and in vivo animal models, go into the same database, together with historical data that all of the partners have, for instance, on mRNA sequencing of beta cells challenged with inflammatory cytokines. This allows us not only to analyze the clinical data, but really to meta-analyze all the data and really find patterns and also novel biomarkers that might allow us to make conclusions of who is at risk for type 1 diabetes and understand this disease. And then work package 5, again, a clinical trial network, but also the potential to do novel ways of clinical trials. Until now, we do clinical trials, 300 people, you randomize them, you blind them, blah, blah, blah. It takes many years. What we want to do is to come to innovative trial design where we have one master protocol where therapy upon therapy can be built in. And this will be done in our clinical centers that have all been, in the last 18 months, accredited. So every center has been visited, they filled in a self-assessment questionnaire, we have visited them and accredited them. You can say, this is trivial, this is boring. Indeed, scientifically it is boring, but it's so essential to know that everybody is taking the blood in the same way, that everybody is doing the OGTTs, the challenge tests, in the same way. So this is the handbook. Now, to me, the most innovative thing is the fact that we have this patient advisory committee. I told you that JDRF was instrumental in creating this call and in really getting FPM members on board and making them willing to participate in this initiative. It is a small market, but what a rewarding market. And so our patient advisory committee, consisting of patients with type 1 diabetes or parents of children with type 1 diabetes, have been there from the beginning. We as clinical researchers have designed the most fantastic trials, the most fantastic ways of collecting samples, and then our patients say, "Are you crazy? I will not participate if you do it so and so." So we have our patients from the beginning involved in the way we design our clinical trials, in the way we design our sample collections, and what is most importantly, and it was mentioned several times, in translating what we do to the public and to the patients. Because it's not only patients, it's the public, it's the taxpayers who need to be convinced that what we're doing is essential. So our patient advisory committee has been instrumental in designing our website, in feeding also into that website. For instance, and Dr. Arnaud from JDRF is sitting here, what they made is a map of Europe, where a patient can hover, or a relative of a person with type 1 diabetes can hover over the map and see what is the center in my area. They can click on it, can get into contact with that center, etc. Crucial part of what we're doing. But what this PAC has also done is made materials, testimonial videos. Also a little cartoon, you can see it there, it's Delta, explaining to children, the 2-year-olds, the 5-year-olds, the 9-year-olds who need to come into the center and give their blood or collect urine and stool. So this little Delta has a video, has a cartoon where it explains to kids what it is like to give blood and to participate in a clinical trial. And so here you can see these videos, the cartoon, also a booklet. They made a booklet with stickers to reward the kids if they come to the clinic, etc. And then social media, of course. I mean, it was mentioned, we can do whatever we want, but we need to show the public what we're doing with their taxpayer money. And so we are trying to be active in Twitter, etc. And again, we as researchers, we're not very used to doing that. So thanks to the PAC, this is now happening, and they're really active and sharing what is happening. And also, in my eyes, a very important initiative is really to show the public what we are doing, what it is, research. And so we organized science days for patients, and this was, for instance, in September in Leuven, where I work, where we invited patients, children, etc., to come into the labs, to come and see what it is really like, what research is doing, and what is happening to their blood and their urine, etc. And so this is also all thanks to the Patient Advisory Committee and the involvement in an IMI 2 program. So INNODIA, we didn't steal the name. It is innovative, I think, in the way we are doing things. We don't do this in splendid isolation. It was already mentioned, we are Europe, but type 1 diabetes is also in the US, is anywhere else. And so, for instance, the CPAT initiative was mentioned by Ian. So I am a member of the steering committee of the CPAT initiative. Because if we find biomarkers, yeah, for FPA, this is crucial that they will be accepted by EMA as surrogate markers of a disease, for instance. So we need to go to EMA. And the CPAT initiative, again supported by pharma companies, is actually doing that for FDA. And so we said, this is too stupid. You're preparing the dossier for FDA. Why don't we work together and do it for FDA and for EMA? And so this is what INNODIA is now doing together with CPAT. We are preparing not only dossiers on biomarkers for FDA, but also for EMA. And also, we are collaborating with other newly starting IMI2 projects, like there is one of the projects, C4C, a project on a generic network on pediatric interventions, clinical interventions in Europe, and they're still not gone through all the stages, but we have told them that if they go through, of course, if it is to do with a disease like type 1 diabetes, we will gladly share our accreditation for clinical centers and what have you. So this is it. I think I stuck to time, Hugh, and I'll come back later for questions, probably. Thank you.
We can take some questions now. So we will have a session after the three presentations, but if there are any pressing questions now, we can take one or two, because you did an excellent job in keeping to time. So. Oh, I think I see somebody through the bright light down there.
At Back of Children's Tumor Foundation. Spectacular. Congratulations. One quick question. So we have been building similar things to you. But we have not been able to gather a lot of the clinical data. And did you encounter any resistance from the clinicians to share their clinical data? And how many IRB walls were pulled up to protect? Okay, thank you. I think we should go for a beer together.
Well,
because
I dye my hair for a reason. Collaborating in Europe, transferring blood from one country to another, ooh la la. Transferring a pancreas from one country to another, ooh la la again. Local IRBs, ooh la la. But I have to tell you, the 17 centers, the 17 clinical centers that are in INNODIA have all gone through IRB approval within a 1-year time span. Yes. So we've worked extremely hard. And again, what was crucial? The patients. Because the patients pushed it through. In some centers, it was needed that they went and talked to the IRB, but they pushed it through. Historical samples, that's a different discussion, because some of the clinicians say, well, these are mine. They got them through public money, but still they say they're mine. So, yes, we need to massage them a little bit, but again, we just had an amendment approved, thanks to the fantastic team of you, because we had the opportunity to get access to historical samples of pancreases that were in Exeter. Exeter is not a partner, so now they become a subcontractor and gave us 120 pancreas samples, which is a unique collection.